Introduction to enzyme mechanisms. An understanding of the complete mechanism of the action of a purified enzyme requires identification of all substrates, cofactors, products, and regulators. Moreover, it requires a knowledge of the temporal sequence in which enzyme bound reaction intermediates form, the structure of each intermediate and each transition state, the rate of the interconversion between intermediate. Number 4, the structural relationship of the enzyme to each intermediate. Number 5, energy contributed by all reacting and interacting groups to the intermediate complexes and transition states. As yet, there is probably no enzyme for which all of these features are completely known. However, many of these mechanetics aspects have been established for enzymes such as chymotrypsin, which will be the focus of our discussion of enzyme mechanism. The structure of chymotrypsin CT. Bovine pancreatic chymotrypsin MR25191 is a protease or enzyme that catalyzes the hydrolytic cleavage of peptide bonds in target protein. CT is specific for peptide bond adjacent to aromatic amino acid residue. Cleavage occurs on the C-terminal side of these residues. The three-dimensional structure of the CT is shown in the figure 6.19 with functional groups in the active site emphasized. The protein consists of three polypeptide chains linked by the disulfide bonds. The three chains are derived by the proteolytic processing of a slightly longer protein known as processing of a slightly longer protein as shown in the figure. The key catalytic residue of the active site are brought together in three-dimensional space in the tertiary structure. The hydrophobic pocket in which the aromatic amino acid side chain of the substrate is bound is highlighted in yellow. Introduction to CT mechanism. The reaction catalyzed by CT illustrates the principle of the transition stabilization and also provide a classic example of journal acid-base catalysis and covalent catalysis. CTX enhances the rate of the peptide bond hydrolysis by a factor of at least 10 to the power raised to 9. It does not catalyze a direct attack of water on the peptide bond. Instead, a transient covalent acyl enzyme intermediate is formed. The reaction has two distinct phases. In the acylation phase, the peptide bond is cleaved and an ester linkage is formed between the peptide carbonyl carbon and the enzyme. The diacylation phase, the ester linkage is hydrolyzed and non-catalyzed enzyme is generated. As covered below, the nucleophile in the acylation phase is the oxygen atom in the side chain of SR195. Proteases with these serine residues that play this role in reaction mechanisms are called the serine proteases. The pKa of a serine hydroxyl group is generally too high for a unprotonated form to be present in significant concentrations at physiological pH. However, in CT, SR195 is linked to HIS57 and ASP109 in a hydrogen bonding network referred to as the catalytic tride. The association of HIS57 and ASP102 raises the pKa of his residue to greater than 12, allowing the HIS to act as an enhanced journal base that can remove the proton from creating an extremely reactive nucleophile. At later reaction stages, HIS57 acts as a proton donor, protonating the amino group in the displaced portion of the substrate.